What is up, party people? We are learning about phylogeny today. So here are our objectives. Feel free to pause and let's get right into it. So phylogeny, however you want to pronounce it, its simplest definition is the relationship of an organism and or a group of organisms compared to their evolutionary history. Phylogenetics is the study of evolutionary relationships, and then the entire field of study is called systematics, which is the field of study that organizes, classifies organisms based off their evolutionary relationships. So evolution of life. We have a few different theories of how did life evolve. One of the most common theories is our endosymbiotic theory, which essentially says that eukaryotes evolved from simpler prokaryotic cells through the formation of symbiotic relationships between one cell engulfing another. So let's break that down a little bit. Symbiotic relationships. That means that they work well together. It's not a parasitic relationship. It's the one where two people meet and they're happy, so they work together. So essentially, there were prokaryotes, there was a baby prokaryote, and there was a big prokaryote. Big prokaryote eats little eukaryote, but they're happy together. So then they take the DNA from the prokaryote and the bigger prokaryote, and then it starts forming something new. That's essentially the theory of endosymbiotic. This is how we explain the mitochondria and chloroplast, because both have their own DNA. So remember, think back to first semester where we talked about how did we get the mitochondria and chloroplast if they have different DNA from the host cell, because all DNA is meant to be in the nucleus. Well, no, these two have different DNA. So this is the theory that is meant to explain how we have that. So with mitochondria and chloroplast, again, that idea is that they were once free living like bacteria cells, and then they were engulfed by a host cell, and then they formed that symbiotic relationship. Now there's a few theories of which organelles came first. There's the nucleus first theory, which is that the nucleus evolved in prokaryotes first, and then it became a eukaryote through the fusion with bacteria that became the mitochondria. The mitochondria theory is that the mitochondria evolved in prokaryotes first, and then obtained the nucleus through additional evolution or fusion. Then there's the eukaryote theory first, which is prokaryotes evolved into eukaryotes, or prokaryotes evolved from eukaryotes and lost their complexity. So essentially, eukaryotes were always here. They always had the mitochondria, they always had the nucleus, but over time they lost their complexity, and that's how we get our non-membrane bound organelles and our prokaryotes that are like dumb versions of eukaryotes. Now, a web of life. A web of life is a complex view that eukaryotes evolved from a pool of ancestors that shared genes through horizontal gene transfer mechanisms. So essentially we have this massive pool, and instead of going from parent to offspring, we're going from friend to friend, and we're passing things along horizontally. Now, horizontal gene transfer essentially is the idea that genetic material is passed from one species to another, not parent to offspring. And if you look to the right, there's some examples of this. Now, a ring of life. Rings of life. They're meant to show all life that arose from a pool of prokaryotes which exchange genes. So essentially we have this pool full of prokaryotes, but the prokaryotes are different. And when they exchange those genes, the new gene combinations created this diversity. That's one of the ideas. The classic model. This is our most common model. This is like the universal one. This is what he wants us to know. Him as in Mr. Mason. It was developed by Carl Linnaeus, and it's based on the morphological characteristics. So it's known as Linnaean taxonomy, and it's our modern system that uses genetic data. So taxonomy is the branch of science responsible for the classification of organisms. And then there's a taxon. We get that from taxonomy. A taxon is a taxonomic level, which is a classification or rank, which is used to essentially characterize the different groups and organisms that have shared characteristics. So we have 8, not 7. If you Google it, it might say 7, but there are 8. Don't get confused. There are 8, and it starts from biggest to smallest. Take a look at the image on the right. We start with domain. We have a bunch of organisms in our domain. Our domain is eukarya, archaea, or bacteria. Below that, we get into the kingdom. Our kingdom is Animalia. And then below that, we have our phylum, and then our class, order, family, genus, species. Each of those is getting more and more individualized. We're not having that diversity as we go down. So again, taxons are ordered from the broadest, so many organisms with most common characteristics, to the most specific, few organisms with very few specific shared characteristics. So again, domain is the largest and broadest, species is the smallest and most specific. So to try and remember this, there is this acronym. Dear King Philip came over for great spaghetti. Domain, kingdom, phylum, class, order, family, genus, species. Back to taxonomy. So how do we characterize animals, species? Well, we use the binomial nomenclature. Bi, two, nomial, name. Two-part name system. So it's our universal two-part naming system that we use to identify species. So this is also known as the scientific name. So we have humans, So our regular common name is a human, but our scientific name is a Homo sapien. And all of these scientific names are in Latin. It is universal across the world that they are in Latin. And the way that we write these is the genus comes first with a capitalized letter on the genus, and then we write the species. The species is not capitalized. Only the genus is, but the entire thing is italicized. So think back to our Zophobos morio. Z was capitalized, the M wasn't, but we italicized the entire thing, and its common name was the superworm. In our example to the right, we have the red panda, which is the common name, and the scientific name is Aurelius fulgens. I don't know. Mr. Mason said that's his favorite animal. It's not mine, but that's okay. Domains of life. We kind of hit on these earlier, but let's get a little bit deeper into them. So domain is the highest and most inclusive taxonomic level. So it contains the largest number of organisms. And we have the 3 domains of life. Oh, this might sound familiar. Yeah, it should. Bacteria, archaea, and eukarya. Take a look at the image to the right, and you're going to see some things that look pretty common. You've seen them before. I hope you have. So bacteria, what is bacteria? It can also be called eubacteria, but it's most commonly called bacteria or true bacteria. So this is common prokaryotes. They lack a nucleus and membrane-bound organelles. They don't have membrane-bound organelles. They also have a peptoglycan cell wall. They can have diverse shapes, so they could come in rods, spears, or spirals. Take a look to the right. We have those spherical little orbs. We have rod legs, which kind of just look like a sprinkle, and then our spirals are the weird little squigglies. Our archaea, or archaeobacteria, are prokaryotes that lack a nucleus and membrane-bound organelles, and they're similar structurally to bacteria, but genetically they are different because archaea are extremophiles. They live in extreme environments, so this could mean a lack of oxygen, extreme heat, extreme pH, extreme salt, extreme pressure. So if we take a look to the right, any of the bacteria that lives within that hot spring, which is filled with sulfur and extreme heat, that's an extremophile. Think about the environment that it's in. And if it seems like an extreme environment, it's going to be classified as an archaea because it's an extremophile. Or if the bacteria lives in a completely dark environment, if it lives at the very bottom of the ocean where the pressure is high and there is no light reaching that organism, it's going to be classified as an extremophile because that's an extreme environment. And eukarya, our favorite, because we're eukaryotes. Eukarya has eukaryotes, which do have a nucleus, and they do have membrane-bound organelles. These can be unicellular or multicellular, but they're complex cellular structures that have complex processes. And this includes the protists, fungi, animals, and plants. Our phylogenetic tree. So a phylogenetic tree, it's essentially a diagram that reflects a hypothetical relationship between organisms and or groups of organisms, and this is based on morphological characteristics and genetics. So we have 2 different types of a phylogenetic tree. Both are shown on the right. Our rooted, which is going to be the top image, is a tree that contains a single evolutionary lineage where a common ancestor is at the base. So at the very bottom, everything stems from this one connected ancestor, and then we branch out as species evolve. An unrooted tree shows relationships among organisms, but it doesn't show a common ancestor, and there could be a number of reasons for this. We might not have the evidence to prove a common ancestor. We might not even have a common ancestor, but we can prove relationships between the more recent organisms. There's a number of reasonings for this, but it doesn't discount either. These are just hypothetical relationships, again, based on the evidence that we have morphologically and genetically. So again, a rooted tree, let's break this down a little bit. So it's branching that indicates hypothesized evolutionary relationships. So a branch point is called a node, and look at the image to the right to see exactly where it's pointing. So essentially we have the main branch, and then we have branches that branch off of that. Where there is that corner, that is a branch node, and it represents where an evolutionary lineage diverges into a new distinct lineage. A clade is all of the members of a branch all the members on the branch. So essentially all of the members of the branch have evolved from a common ancestor. So if you look at the second branch point, D and C, that's a clade. B and A, that's a clade. E, even though it's alone, is still a clade. And then there's also sister taxa, and this means that the lineages have that branch. These are lineages that branch from the same point and node. So essentially it branches from a node, and then that branch branches off again. And the 2 branches are sister taxa. So D and C are sister taxa, and B and A are also sister taxa. Now, the phylogenetic tree is kind of a ladder-like structure. Again, you can think of it as a tree, a ladder, a dichotomous key, whatever works for you. But it's a ladder-like phylogenetic tree, and it has vertebrae, and it's rooted by an organism that lacks a vertebrae column. So again, think of the dichotomous key. Vertebral column, yes or no. Hinged jaw, yes or no. Legs, yes or no. And each of these characteristics is where we hit the evolutionary points. So at each of the branching points, the organisms have a different characteristic, and they're placed in different groups based on the characteristics that they share. This is the morphology. Unless it's specified, the length of the branch does not indicate an amount of time has passed. So essentially, it could be a really long branch, but that doesn't mean that it's been 10,000 years. It doesn't even indicate a time. It's simply that whoever drew the phylogenetic tree just drew that branch really long. So again, at each breaking point is that characteristic that differentiates the previous from the new. A cladogram. This is our other version of our trees and classification. So this is a simplified phylogenetic tree, and it's based on morphology, so physical characteristics. And again, we have clades on the cladogram, and these may or may not correspond with those on a full phylogenetic tree. It varies. And again, the cladograms are based on morphology, so physical structures. So these are going to be homologous traits that are shared and shared characteristics. So it could be shared ancestral characteristics, so those which represent all members of a tree. And it could also be shared derived characters that are derived at some point in evolution, but that doesn't include all ancestors on the tree. The issue with this is that we could say that, say, the forum of a human and the forum of a whale, we could put them closely related together, even though they're really not, because of homologous versus analogous structures. So there is that differentiation there. And so that's why we include genetics in the full phylogenetic tree, because using genetics, we can actually prove with DNA and amino acid sequencing whether or not something is truly related genetically through evolution, rather than saying, oh, these look similar, therefore they must be similar. That's why we have the differences. So systematics, which we talked about earlier, but it's our classification, our entire field of study, it uses multiple sources to determine the possible evolutionary relationships. So morphology, form and function, homologous structures, analogous structures. Homologous structures are structures that have similar functions and they're derived from a common ancestor. Analogous structures, same function, similar structure, are not derived from a common ancestor. They derived similarly due to the environments that they are within, not because of an ancestor. So again, these things can cause species to look related when they're not, and it could lead to inaccurate cladograms and phylogenetic trees. We also look at behavioral characteristics and physiology. Now on to modern genetics. So modern genetics increase our understanding of possible evolutionary relationships. It's changed existing trees because of the DNA evidence that shows closer relationships than previously understood. So what we might have had in the 1900s, that tree might and likely does look very different from a tree that we have today in 2026 because of the technology and advancements we've made in the fields of genetic identification. And also, it's limited due to just 4 nucleotides, which can result in false relationships due to similar sequences and mutations. And mutations can mask the actual relatedness since they can change the sequence. There are a lot of limitations to this as well. There are several areas which limit the construction of phylogenetic trees because they cause confusion when determining actual relatedness between organisms, homologous versus analogous structures, DNA limitations, mutations that change the DNA sequencing, incomplete genomic records, gaps in fossil records, and the significant number of species. There are more than 2 million and as many as 100 known species. That is so many. So it creates a lot of difficulty. Now we do have 2 different distinctions. We have maximum parsimony. This is the tree with the fewest hypothesized evolutionary changes, including mutations. And it's the theory that the one with the fewest evolutionary changes is most likely to be accurate. And this comes from Occam's razor, who again had this theory that the simplest explanation is most likely correct. So go with the simplest explanation. Whereas maximum likelihood is a statistical approach that identifies the trees most likely to produce relationships through DNA data based on probability rules of how DNA changes over time. So there's 2 different ways that we can look at phylogenetic trees and hypothesize which one is more accurate. Hope you guys enjoyed this video.